Welcome to our video about the fascinating world of Pokemon and their placement on the Tree of Life. Today, we take a closer look at Kingler and its relationship with all insect Pokemon as they are all part of the Arthropods clade. Ready? Let's go! Arthropods are a phylum of invertebrate animals. They include insects, other hexapods, myriapods such as centipedes and millipedes, crustaceans, e.g., crabs, barnacles, arachnids, e.g., spiders, scorpions, mites, other chelicerates, and the extinct trilobites. Arthropods are an extremely successful phylum. Around 80% of all known extant, non-fossil, animal species are arthropods. These include nearly 1 million insect species, about 10,000 other hexapods, approximately 16,000 myriapods, over 100,000 arachnids, and nearly 50,000 crustaceans. These are rough estimates. Exact numbers vary depending on the source and year of publication. In Germany alone, more than 39,000 species of this highly diverse phylum have been documented. Arthropods likely originated during the Cambrian explosion, around 540 million years ago, when multicellular animal phyla suddenly appeared in the fossil record. Their closest relatives are the tardigrades, Tardigrada, and velvet worms, Onychophora, with which they form the group Panarthropoda. Some taxonomists refer to this broader group as Arthropoda, while the true arthropods are then called Euarthropoda. In traditional systematics, based on morphology, this group was classified together with annelid worms as part of the Articulata. However, more recent molecular data suggest a closer relationship between arthropods and nematodes, Nematoda, and their closest relatives, Cycloneuralia. This combined group is known as the ecdysozoa, or molting animals. The body covering of arthropods forms an external skeleton, exoskeleton, which is secreted by the epithelial cells of the outermost cell layer, epidermis. This is called the cuticle. Because of this rigid outer shell, growth is only possible through periodic shedding and replacement of the cuticle, molting. The hardened outer shell of the body and limbs is the most distinctive feature of arthropods. Similar structures are found only in their close relatives, the tardigrades. The cuticle consists of chitin and numerous proteins. Chitin and proteins are cross-linked by dopamine-derived compounds in a process called sclerotization. The cuticle can remain flexible, e.g., in the abdomens of most spiders or many insect larvae, but typically contains hardened, more heavily sclerotized plates, sclerites, which are separated by less sclerotized joint membranes or, in some cases, fused into rigid shells or armor. In many crustaceans, myriapods, and a few insect larvae, the cuticle is further reinforced by calcium deposits. The cuticle is colorless, straw-colored, or yellowish, but often appears dark or colorful due to melanin, pigments, or structural coloration. In insects and spiders, the outermost layer of the cuticle includes a lipid layer that prevents water loss. This layer is absent in myriapods and all terrestrial crustaceans. Arthropods have a segmented body. In the basic body plan, each segment has the same structure with similar organs, appendages, etc., except for the first and last segments. In all arthropods, this basic plan is modified to varying degrees, but the segments are usually clearly distinguishable, and many organs, e.g., limbs, heart openings, glands, muscles, are arranged segmentally. Traditionally, it has been assumed that the basic body plan of arthropods originally featured an indefinite number of internally similar segments, with the number becoming fixed only later through specialization. This assumption is largely based on analogy with annelid worms. However, all arthropods, except for some myriapods, branchiopod crustaceans, and the extinct trilobites, possess a fixed number of segments. Even the lobopodians from the Cambrian fossil record, generally considered stem group representatives, had a fixed, and relatively small, number of segments. There is still debate among researchers as to whether segmentation is a primitive trait inherited by arthropods from the common ancestor of bilaterians, or whether it is an evolutionary innovation. If the latter is true, segmentation in other segmented animal phyla, especially annelids and chordates, would have arisen independently through convergent evolution. Each segment may bear a pair of jointed appendages, legs, mouthparts, antennae, although these may be absent on certain segments. The developmental basis of body segments and limb segments is comparable. Appendages can be viewed as secondary body axes. The nervous system is also segmentally organized, consisting of a double nerve cord running along the ventral side, with one ganglion and a transverse connection, commissure, per segment. This is known as a ladder-like nervous system. A dorsal heart and an open circulatory system are also typical features. The ancestral structure of a segment consists of a trunk portion with a ganglion and a pair of biramous, two-branched, appendages, known as biramous limbs. The inner, ventral, branch is called the endopod or walking leg, while the outer branch is the exopod or gill branch, indicating their respective functions. As arthropods diversified over evolutionary time, segments began to group or fuse into functional units called tagmata. The specific segments that fuse into a tagma vary among different arthropod groups. In all arthropods, some of the anterior segments have fused to form the head, which contains a brain composed of fused ganglia and appendages modified into antennae and mouthparts, either uniramous or biramous. The number of segments incorporated into the head differs across arthropod groups. In primitive arthropods like the Remipedia, the body behind the head still consists largely of nearly identical segments. Trilobites also had a similarly repetitive body structure. In myriapods, species with numerous similar segments evolved secondarily from ancestors with relatively few segments, typically 12 to 15, depending on the lineage. In millipedes, Diplopida, two consecutive segments have fused into diplosegments, each bearing two pairs of legs. In many centipedes, a broad and a narrow segment alternate along the body. In insects, tagmatization is more advanced, multiple segments have fused into the head, thorax, and abdomen. These three body regions serve very different functions. The appendages of the original head segments have been transformed into antennae and mouthparts. In the thorax, three segments have fused into a tagma, each bearing a pair of uniramous limbs that have lost their outer branch and serve as walking legs. These limbs are called uniramous. In the abdomen, appendages may be completely reduced or transformed into specialized structures, e.g., gills, reproductive organs, ovipositors, but they never function as walking legs. In arthropods that lack visible gills, respiration occurs either through invaginated gills, lungs, or through branched invaginations of the body wall known as tracheae. The abdomen may consist of varying numbers of limbless segments. Insects' wings are traditionally not considered limbs but rather modified lateral plates of the thoracic segments. However, recent genetic and developmental studies suggest that wings may derive from the gill branch of the ancestral biramous limb. In this case, the gills of aquatic insect larvae, such as those of mayflies, are homologous structures. Fossil insect larvae from the Mesozoic era exist that appear to represent transitional forms. In other arthropod subphyla, segment fusion has occurred in different ways. Spiders consist of two main body sections. The anterior section, prosoma, is composed of 6 fused segments. One pair of limbs has evolved into mouthparts, chelicerae, 1 into pedipalps, and 4 into walking legs. The limbs of the posterior section, opisthossoma, have lost their walking branch. The gill branch is present as book lungs, which are invaginated into the abdomen. In some species, these book lungs have evolved into tracheae. The last pairs of limbs have transformed into spinnerets. In harvestmen and pseudoscorpions, the front and rear body sections are fused into a compact body.
In horseshoe crabs, Xiphosaura, the abdomen still bears limbs, leaf-like appendages, whose gill branches form book gills, structures very similar to the book lungs of spiders. In crustaceans, the body is usually divided into head, thorax, and abdomen, but this basic plan has been modified in many ways. In several crustacean orders, 1 or 2 original thoracic segments have functionally fused with the head. The thorax may have a highly variable number of limb-bearing segments. A limbless abdomen may be present or absent depending on the lineage. In Malacostraca, the abdomen is replaced by a pleon that bears limbs. At the end of the body, they have a tail fan formed from several fused appendages. Many other crustaceans have long appendages here, usually called the furcae.in. Many crustaceans, body segmentation may be obscured by a body duplication called the carapace. This can be a single piece or a bivalved structure with movable halves. In some groups, e.g., Ostracoda, Conchostraca, the carapace encloses the entire body, giving it a shell-like appearance. In Malacostraca, the carapace, originally attached only to the head, is fused with the thorax, forming a rigid, immobile cephalothorax. In some sessile groups, such as barnacles and goose barnacles, and especially in parasitic groups, the body structure can be so radically transformed that the animal is barely recognizable as an arthropod. Arthropods possess a variable number, up to 9, of eyes on the head, with highly diverse structures and functions. However, all arthropod eyes are based on a few fundamental types, and even their highly modified forms can often be connected through intermediate stages. This suggests that all arthropod eyes are homologous structures. Arthropod eyes can be grouped into two main types, distinguished by their innervation, receptor, and transmitter molecules: lateral eyes and median eyes. The laterally positioned eyes are compound eyes, consisting of many individual units called ommatidia. In insects and crustaceans, the structure of ommatidia is highly uniform. Each contains a retina with 8 light-sensitive sensory cells, topped by a transparent crystalline cone formed by 4 so-called Semper cells. Flanking this are 2 pigment cells that shield the ommatidium from lateral light, and also secrete the outer cornea. In most arthropod groups, the cornea is thickened into a lens-like shape. In addition to the 2 so-called primary pigment cells, there are numerous secondary pigment cells. The structure of ommatidia can be secondarily modified or simplified in other arthropod groups. Compound eyes are present in most insects, but absent in other hexapods, and in many crustaceans. Among chelicerates, compound eyes are found only in one group, the horseshoe crabs. The only myriapods with compound eyes are the house centipedes, Scutigyromorpha. However, the ommatidia of these groups differ significantly in fine structure from those of insects and crustaceans. For example, in the horseshoe crab Limulus, each ommatidium consists of more than 300 cells. In groups where compound eyes are present, they are typically the primary visual organs. The photoreceptor cells of all retinula cells within an ommatidium work together to form a single image point. The resolution of a compound eye therefore depends on the number of ommatidia, which can range from just a few to several hundred, e.g., about 700 in the fruit fly or 1,000 in the horseshoe crab, and up to a maximum of 30,000 in some species of large dragonflies. The spatial resolution of a compound eye never matches that of the highly developed lens eyes of vertebrates and cephalopods, but their temporal resolution, especially for detecting motion, is significantly better. In addition to spatial vision, many arthropods across all lineages can distinguish colors. As in humans, their color vision is based on the presence of different visual pigments, typically opsin variants, that respond to different wavelengths. Also similar to humans, most arthropods possess 3 such pigments, enabling trichromatic vision. However, unlike humans, their sensitivity is often shifted toward the shorter wavelengths of the spectrum. As a result, many species cannot perceive red, but can detect ultraviolet light. Another sensory capability in which arthropods surpass humans is their ability to perceive the plane of polarization of light. This allows them, for example, to determine the position of the sun even under overcast skies, and helps them detect bodies of water. Almost all groups with compound eyes also possess additional eyes derived from the median eyes, with the exception of myriapods, which lack median eyes entirely. In other arthropods where the eyes are not completely reduced, additional eye types are also present. In insects, 3 simple eyes, ocelli, are typically located on the forehead. Their exact function is not fully understood, but in some groups they are known to assist with orientation during flight. In many crustacean groups, 3 median eyes, for amphilopods, form a central visual organ known as the nauplius eye. This is the primary sensory organ in crustacean larvae, and is also retained in many adults. In malacostricans, there are often one pair of dorsal and one pair of ventral median eyes, though other numbers may occur in different groups. True spiders, which never possess compound eyes, have only one pair of median eyes and usually 2 or 3 pairs of lateral eyes. However, these lateral eyes are never used for image-forming vision. In wolf spiders, jumping spiders, and crab spiders, they function as motion detectors. In other groups, they are used for spatial navigation based on the position of the sun, often by detecting polarized light. Within the highly diverse arthropod phylum, many groups have evolved specialized adaptations that enable unique sensory capabilities. For example, jumping spiders have enlarged median eyes that allow them to form images using principles similar to those of vertebrate lens eyes. Some groups, such as the copepod Lepidosira, or the larvae of the diving beetle Thermonex, have developed eyes with a minimal field of view and high resolution, enabling them to scan their surroundings like a reading head using scanning movements. Adaptations to low-light environments are especially widespread among arthropods. Many species that are active at dusk or live in deeper ocean zones have modified their compound eyes into a form where light from a broader portion of the environment can reach each photoreceptor unit. This is known as a superposition eye. However, some primarily ground-dwelling groups have completely lost their eyes. This results in significant energy savings, as in all arthropods capable of image-forming vision, a substantial portion of the brain is devoted to visual processing in addition to the eyes themselves. In addition to eyes, many arthropods possess a skin, or nerve-based light sensitivity, that is entirely independent of having eyes. This can serve, for example, in predator detection through shadow perception, as in the crayfish Procambarus clarkii, or in regulating gonad development via day-length detection, as seen in some butterfly species. Arthropods primarily use 2 mechanisms to perceive mechanical stimuli: sensory hairs or bristles, and receptors that detect deformation of the cuticle, the exoskeleton. These organs are used for proprioception, awareness of body position, close-range orientation, touch, and long-range orientation, vibration, current detection, and hearing. While many sensors can perform multiple functions, some are highly specialized for specific tasks. The sense of touch in arthropods largely relies on simple sensory hairs. In addition, especially among air-dwelling insects and arachnids, extremely fine sensory hairs have evolved to detect air currents and vibrations. These are called filament hairs in insects and trichobothria in arachnids. Unlike ordinary hairs, they are typically mounted in a ring-shaped depression of the cuticle. Embedded directly in the cuticle of all arthropods are sensilla, which can detect both tactile stimuli and vibrations. The receptors, scolopidia, can form specialized sensory organs, such as the slit sense organs in arachnids, which in true spiders are grouped into lyre-shaped organs. Chordotonal organs, found only in insects and crustaceans, are not visible from the outside. Their scolopidia consist of 1 to 4 sensory neurons, a sheath cell, scolopale cell, several attachment cells, and surrounding glial cells. Their exact function is not fully understood, but it is known that modified cilia are responsible for stimulus detection. In insects, additional dome-shaped campaniform sensilla are present. Important organs for touch, vibration, and current detection are the antennae. A specialized organ at the base of the antenna, the Johnston's organ, is responsible for sensory perception. Spiders and many insects perceive gravity through the tension exerted on the body suspended by the legs. In spiders, this is mediated by lyre-shaped organs. In some insects, the Johnston's organ detects gravitational pull on the antennae. Aquatic crustaceans often have little density contrast with their surrounding medium. They can float in water. As a result, they have developed their own gravity-sensing organs called statocysts. These consist of a capsule lined with numerous inward-facing hair cells. During molting, grains of sand are introduced into the capsule from the outside. These grains press on the hairs, indicating the direction of gravity. The function of statocysts is nearly identical to the gravity-sensing organs of vertebrates. Statocysts are usually located in the basal segments of the antennae, but in some groups, they are found at the rear of the body in the Arthropods or telson. The perception of sound waves is a specialized form of vibration sensing. Many arthropods use the same hair sensors to detect air movement, e.g., from an approaching predator, or ground vibrations. In some true spiders, sound perception via trichobothria has been demonstrated. In mosquitoes, males recognize females by the sound produced by the rapid vibration of their wings, a sound that humans can also hear. In these males, the antennae are greatly enlarged, and the Johnston's organ serves as the hearing organ. Acoustic communication through vibrations detected by trichobothria has also been demonstrated in a whip scorpion and may be widespread. In insects, in addition to these close-range sensors that detect particle movement directly, specialized organs have evolved that also respond to pressure differences. That is, they can perceive sound waves in a manner similar to vertebrate ears. These are called tympanal organs. Beyond predator detection, sound perception is used by sound-producing insects such as grasshoppers and cicadas, and possibly water bugs, for mate finding. Parasitic flies have exploited this by evolving tympanal organs capable of detecting cricket songs, allowing them to locate hosts on which to lay their eggs. Most insects with tympanal organs use them for a single purpose: to detect the ultrasonic echolocation calls of bats. This ability has evolved independently in 7 families of moths, in mantises, green lacewings, and 2 beetle groups, tiger beetles and scarab beetles. Tympanal organs are modified chordotonal organs, in which the sensory cells are located on internal air sacs, expanded tracheae. Their position varies greatly: in the abdomen, cicadas; most butterflies; shorthorned grasshoppers; tiger beetles; in the thorax, owlet moths; mantises; in the legs, longhorned grasshoppers; at the wing base, hawk moths; lacewings; behind the head, flies; scarab beetles; or on the mouthparts, hawk moths. While many groups can only hear a narrow frequency band in the ultrasonic range, some possess highly developed directional and distance hearing. Only in recent years has it been confirmed that crustaceans, Decapoda, also possess a sense of hearing. They can detect low-frequency sound waves from about 100 to 3000 Hz using their statocysts, chemical sensory organs for short-range orientation, taste, and long-range orientation, smell, are present in all arthropods in the form of a wide variety of sensillum types. In groups lacking highly developed eyes, these are typically the most important organs for locating food. Additionally, social communication and mate finding in many arthropods rely on pheromones, which are detected by chemical sensory organs. The primary organs for chemical orientation are usually the antennae, but also frequently the palps and the distal segments of the legs. In species lacking antennae and palps, such as whip spiders, the legs can be richly equipped with receptors. Many arthropods also have chemical sensors scattered across the entire body surface. On a single arthropod antenna, one can regularly find hundreds of thousands of chemoreceptors. The sensilla on the antennae are typically divided into 2 morphological groups: hair sensilla and pit sensilla, with transitional forms, peg or cone-shaped projections within a pit. Additionally, various hair types are distinguished based on their shape, including dome-shaped campaniform, flask-shaped ampullocele, cone-shaped basiconic, and several others. Although the shape of a receptor often correlates with its function, it has been shown that similarly shaped sensory organs can serve different functions in different animal groups. A common feature of all chemoreceptors is that their surface is covered with pores in the micro to nanometer range. These pores connect the hollow interior, lumen, of the sensillum with the external environment. Molecules that diffuse and are detected by specialized nerve cells, which serve as the actual sensory cells, very often a single sensory hair can process multiple types of stimuli, for example, as a combined chemo and mechanoreceptor, and in rare cases, even as a trimodal receptor. On a single antenna, 5 to 10 different types of sensilla are typically distinguishable based on their morphology. Sensilla involved in food acquisition usually have a relatively broad, but highly differentiated sensory spectrum across different cells, allowing them to detect a wide variety of soluble or volatile substances such as amino acids, peptides, sugars, nucleotides, or amines. From the specific activation patterns of various sensors, the corresponding substance can be identified, similar to how the human sense of taste works. In addition, there are highly specialized sensors for signaling substances, pheromones, that respond exclusively to a single compound. Several arthropod species, including butterflies, mosquitoes, woodlice, millipedes, and ticks, possess specialized sensors that specifically respond to carbon dioxide. These are usually sensilla on the antennae, often flask-shaped, but some species, such as millipedes, have also developed specialized sensory organs on the surface of the head. The biological function of this sensory ability includes host detection in blood-feeding species, or avoidance of suffocation in soil-dwelling species that live in confined spaces. Although this is not strictly a chemical sense, arthropods perceive it using similarly structured sensilla. Cone-shaped sensilla often serve both functions, sometimes even in combination. The specialized infrared radiation detectors of the fire beetle, Melanophila, have become famous. These beetles can detect forest fires from up to 12 kilometers away, and fly directly toward them to lay their eggs in the charred wood. Magnetic field perception is widespread in the animal kingdom, and has also been demonstrated in many arthropods. It is mediated by intracellular iron oxide particles, which have been found in social insects and crustaceans, for example. The magnetic sense likely serves spatial orientation over large distances in all cases. While it was once believed that arthropods lacked the ability to sense electric fields, this has now been disproven. Crustaceans have been shown to possess such a sense. However, their sensitivity is an order of magnitude lower than that of the highly developed electroreceptors found in many fish species. The exact function of this sense in crustaceans is still unclear, but it may play a role in spatial orientation or in detecting prey. In many arthropod species, males and females can only be distinguished by their reproductive organs, but in others, the sexes differ significantly in external appearance. One example of pronounced color differences between the sexes is the large gold grasshopper Chrysochrone dispar from the order Orthoptera. Another striking example of sexual dimorphism is the stag beetle Lucanus cervus from the order Coleoptera. In this species, males have greatly enlarged, antler-like mandibles, which they use to fight each other for access to the less conspicuous females. In some species, dwarf males exist that are significantly smaller than the females, for example, in the pale tussock moth Euproctus similis from the order Lepidoptera, or the wasp spider, Argiope bruennichi from the order Araneae. There are many cases in insects where males are winged while females are not, such as in the common cockroach, Blatta orientalis, from the order Blattidae, or the rusty tussock moth, Orgia antiqua, a species of moth. In extreme cases, the female remains larva-like and relatively large, while the male is fully developed, capable of flight, and possesses significantly more advanced sensory organs. An example of this is the small moth Heterogenes pinella, also from the order Lepidoptera. Arthropods have evolved various strategies to attract mates. In many butterflies, females have simple, thread-like antennae, while males have comb-like antennae that provide a larger surface area to detect the female's sex pheromones from a greater distance. Other species use visual signals. Male fiddler crabs wave their enlarged claw, and fireflies attract mates with species-specific flashing patterns. Sounds are also used. Wolf spiders court with tapping signals. Giant crab spiders attract females by vibrating the surface they sit on, and longhorned grasshoppers produce chirping sounds through stridulation. Parthenogenesis is fairly common in arthropods. It occurs in some mites, acari, crustaceans, crustacea, and insects. Examples include the great green bush cricket and the summer generations of aphids, which produce winged sexual forms in autumn. Hermaphroditism is a rare exception in arthropods. In species like Cyatra carinata, Isopoda, and many Tanidaceans, Tanidacea, individuals are first female and later become male, protogynous hermaphroditism. In terms of brood care, arthropods display the full range, from laying eggs in open water with no parental care to live birth, as seen in Euscorpius mesotrachus. Insects often lay their eggs on host plants. Endoparasites like chalcid wasps and the common oak gall wasp lay their eggs directly into the host. Female cave spiders and cellar spiders of the genus Phalcus wrap their eggs in silk and carry the egg sac with them. The female noble crayfish carries her eggs glued to her body until they hatch. Some species, such as earwigs, guard their eggs in a protected burrow until they hatch. Isopods have a brood pouch similar to that of marsupials, in which they carry their young. This is seen in species like the common woodlouse and aquatic isopods. The spotted tree bug not only guards and protects her eggs, but also leads her young to food in a line, like a duck with ducklings. Some species, such as the common earwig, cobweb spiders, and most eusocial insects, go even further in their brood care and feed their young. In arthropod eggs, embryonic development begins with total cleavage in yolk-poor eggs and superficial cleavage at various points on the surface in yolk-rich eggs. In superficial cleavage, a spherical single-cell layer forms around the yolk, often with many nuclei but no cell boundaries, a syncytium. Next, the cells migrate to one side of the yolk and form a germ disc, from which the embryo develops. Only in primarily aquatic groups does a primary larva hatch from the egg. This includes most crustaceans and the sea spiders, Pycnogonida, among the chelicerates. Crustaceans hatch as nauplius larvae with 3 limb-bearing segments, those of the first and second antennae, and the mandibles, while sea spiders hatch as protonymphon larvae with 3 segments, precursors of the cheliphores, palps, and ovigerous legs. With each subsequent molt, additional segments are added from a posterior growth zone. The limbs of these segments often begin as unsegmented buds and only gradually develop into functional appendages through further molts. Even the limbs of already existing anterior segments are remodeled during later molts. In some cases, they are resorbed, i.e., present in larvae but absent in adults. In crustaceans, the animal may pass through several anatomically and functionally distinct larval stages with different lifestyles over a long developmental period involving many molts. This ancestral arthropod developmental pattern, in which the adult segment count is gradually reached, is called anamorphosis or anameria. In the most primitive case, exactly one segment is added with each molt. However, in many arthropods with anamorphic development, several segments can be added at once. In numerous groups, this ancestral pattern is modified such that the early developmental stages are completed within the egg, so the first free-living larval stage is already more advanced in development. Many free-living nauplius larvae of crustaceans, for example, do not feed. That these are modified forms can be indicated, for instance, by the embryo secreting a cuticle at the corresponding stage. Eventually, in many groups, it has independently occurred that all larval stages are completed within the embryo, or development has become so modified that no trace of these stages remains. In such cases, a juvenile hatches from the egg that already closely resembles the adult in form. This is the norm among terrestrial arthropods, since nauplius or protonymphon-like stages would not be viable on land. This also applies to terrestrial isopods, which are crustaceans. If the first free-living larva already possesses all the segments of the adult, this is referred to as telomorphosis or telomeric development. Development without a primary larval stage is called direct development. Larvae that already largely resemble the adult in organization are also seen in the primarily marine horseshoe crabs, which belong to the Chelicerata. Direct development is typical, for example, of all true spiders and the more primitive hemimetabolous insects. In millipedes and in the protoruns, Protura, which belong to the hexapods, the hatched animal closely resembles the adult in form and lifestyle, though additional segments are added during later molts. A completely modified form of development characterizes the holometabolous insects. In these, a larval stage has secondarily evolved that differs entirely in form and lifestyle from the adult imago. The origin of these larval stages is not fully understood. They may derive from a stage known as the prolarva, which in many hemimetabolous insects occurs between the embryo and the first true larval stage. The tissue of the holometabolous larva is largely broken down during a resting stage, the pupa, and the adult is newly formed from bud-like structures called imaginal discs. Species that hatch with only a few segments and complete their segment count post-hatching anamorphosis include horseshoe crabs, Xiphosuera, and sea spiders, Pycnogonida. Among the Chelicerata, most millipedes, Myriapoda. Except epimorphic centipedes, Protura. Among the hexapods, Hexapoda, species that hatch with the full number of segments, telomorphosis, include nearly all Chelicerata insects, Insecta, Dipleruns, Diplura, and springtails, Collembola. Among the hexapods, Scolopendromorphs, giant centipedes, and geophilomorphs, sol centipedes. Among the centipedes, Chelopoda. In insects, all transitions can be found between forms that establish all or nearly all segments simultaneously during embryogenesis, long germ, and those in which anamorphosis is retained during embryonic development. Within the egg, short germ, most arthropods are small animals. Although their size range overlaps with that of vertebrates, they are generally much smaller. What limits the body size of these otherwise highly diverse and successful animals has long been debated. One theory suggests that the primary limitation lies in the exoskeleton. While an exoskeleton-based body plan is theoretically capable of supporting large sizes, especially since deep internal enfoldings, called apodemes, serve as muscle attachment points, the weak point is the molting phase. In particular, large terrestrial arthropods would literally collapse under their own weight during molting. Another theory points to the open circulatory system and the comparatively less efficient respiratory system as the main constraints. Supporting this view is the fact that giant insects with significantly larger bodies than modern forms appeared during periods of the Paleozoic era, when atmospheric oxygen levels were much higher than today. Terrestrial arthropods breathe through air-filled tubes, tracheae, and while blood is not irrelevant for oxygen transport, it plays a much smaller role than invertebrates. Contrary to some assumptions, air transport in the tracheae is not purely passive. Arthropods can actively compress the tubes to accelerate respiration. Nevertheless, increasing body size likely requires a disproportionately large increase in tracheal diameter, especially noticeable in the legs. Eventually, there may not be enough space left for other vital organs. At the same time, the open circulatory system cannot support a significant increase in blood pressure, making it difficult to optimize oxygen transport via the blood. A third theory proposes that the unmyelinated, naked nerve fibers of arthropods limit reaction speed at larger body sizes. However, myelin sheaths have been found in crustaceans, and arthropods possess giant nerve fibers that can transmit signals for vital movements, e.g., escape reflexes, at speeds comparable to vertebrate nerves. It's also possible that the presence of vertebrates has made large body size ecologically disadvantageous for arthropods, as the available ecological niches are already occupied. Moreover, increasing body size does not come without trade-offs: longer development times, and thus longer generation intervals, higher food requirements due to increased metabolism and resting energy expenditure, and a greater risk of predation. Whether there is an optimal body size for a given body plan, and where that might lie for arthropods, remains an open question in science. However, it is notable that there are more medium-sized arthropods, in the centimeter range, than very large or very small ones. The largest known living arthropod is a marine crab, the Japanese spider crab. The coconut crab, Birgus latro, is the largest terrestrial arthropod, reaching up to 40 centimeters in body length and weighing up to 4 kilograms. Extremely small arthropods, around 0.1 millimeters, are found among mites, springtails, and copepods. The smallest insects, parasitic wasps, measure just 0.15 millimeters. In these miniature forms, many organ systems are reduced, and body and limb segmentation is often indistinct. Arthropods inhabit nearly all known environments and occupy a vast array of ecological niches. Arthropods can therefore be considered the most successful animal phylum on Earth. The earliest arthropods, like the trilobites, lived in the sea. Among the extant, living groups, crustaceans have the highest number of marine representatives. Within the chelicerates, sea spiders and horseshoe crabs are entirely marine, while nearly no other chelicerate species inhabit the ocean, with the exception of a few marine mites. Neither myriapods nor hexapods have produced truly marine representatives. A few insect species, such as the non-biting midge clunio, have only managed to colonize intertidal zones. Numerous crustaceans also live in freshwater, and some groups, such as the brachiopods, are even more species-rich in freshwater than in marine environments. Among the chelicerates, mites have secondarily colonized freshwater with the water mites, meaning their ancestors were terrestrial. Likewise, the many freshwater-dwelling insect species also descend from land-dwelling ancestors. This is evident, for example, in their use of a tracheal system for respiration, whereas primarily aquatic crustaceans breathe through gills. Many aquatic insect larvae, such as those of mayflies or damselflies, have secondarily developed gills again, but these are penetrated by air-filled tracheae. Many insect larvae absorb oxygen directly through the skin, and some possess simple tubular gills filled with hemolymph to increase surface area. In fact, many freshwater arthropods breathe air. They possess a tracheal system with open spiracles. Some can refuel air at the water surface using snorkel-like breathing tubes, such as the larva of the rat-tailed maggot, larva of the drone fly. Others live in an air bubble, like the diving bell spider, or trap a layer of air against their body surface, like the great diving beetle. Most insects, nearly all arachnids, and all myriapods are terrestrial. Among crustaceans, some lineages have independently made the transition to land. The best adapted to terrestrial life are the terrestrial isopods, while land-dwelling decapods, such as land hermit crabs and land crabs, and amphipods must return to water at least for reproduction. Arthropods, alongside vertebrates, are the only group in the animal kingdom to have produced organisms that walk on legs, that is, they do not merely push themselves forward but use true locomotion on limbs. The function of arthropod legs is fundamentally the same in both aquatic and terrestrial forms. For example, it is not possible to determine whether a fossil species was aquatic or terrestrial based solely on its walking legs. Only a few groups have secondarily lost their legs, and these are predominantly parasitic forms. In many crustaceans, the limbs serve dual functions. They are used both for locomotion and for feeding. In more anatomically derived, modern forms, these functions are usually separated. That is, different limbs are specialized for one function or the other. Most aquatic lineages have produced swimming forms. Swimming organs are typically modified and specialized appendages. Some crustacean lineages have independently evolved leaf-like limbs, philopodia, that are no longer suitable for walking. Forms that belong to the zooplankton, such as certain crustaceans, mites, and insect larvae, are continuous swimmers. The development of wings was of exceptional importance for the radiation of insects. The acquisition of flight was likely the key innovation that made flying insects the most species-rich group of organisms on the planet. In contrast to continuous swimmers, there are no continuous flyers. However, many dragonfly species, for example, cannot walk with their legs. Their legs are used exclusively for grasping and feeding. It is often overlooked that web-building spiders have, in a sense, also developed a form of flight. The dispersal of especially young spiders via silk threads they release themselves, known as ballooning, allows them to rapidly colonize suitable habitats. Some lineages have evolved unique modes of locomotion. Many species, such as jumping spiders, fleas, and springtails, are excellent jumpers. Rare are forms that crawl like snails, such as the larvae of the slug moths. Many crustaceans, most spiders, and various insects are predatory. Numerous species feed on blood, such as the deer ked on deer, the red poultry mite on birds, fish lice on fish, or the varroa mite on the western honeybee. The larva of the red velvet mite attaches to insects in a manner similar to the castor bean tick, Ixodes ricinus, on humans. Mite infestations in the ear can lead to ear mange. Follicle mites live in the hair follicles of mammals, and feather mites live on the feathers of birds. Many of these species are harmless to their hosts, while others cause mange. Botflies live in the stomach, nasal and throat cavities, and/or lungs of mammals. Ichneumon wasps are internal parasites that ultimately kill their host, which is why they are referred to as parasitoids. Various species such as spider mites and aphids feed on plant sap. Many butterflies, honeybees, and other animals feed on the nectar of flowering plants. Caterpillars of butterflies and larvae of beetles feed on plant leaves. Gall wasps lay their eggs inside plant tissues. Eusocial insects have complex chemical communication systems, which some species exploit to infiltrate the colonies of other insects without being attacked. The death's head hawkmoth feeds on honey directly from honeycombs. Bees likely do not attack it due to its chemical camouflage. The ichneumon wasp, Ichneumon humerus, is a hyperparasite. It parasitizes the mountain alcon blue butterfly, Maculinea rebeli, which itself parasitizes ants. Some ants cultivate fungi, like the leafcutter ants, or keep aphids as livestock, as seen in the redwood ant Formica rufa, or the black garden ant Lasius niger. Ants have a wide range of fascinating relationships with blue butterflies, ranging from mutualism to parasitism. Fossils of stem group representatives of arthropods have been found dating back to the early Cambrian. Their actual origin likely lies in the late Precambrian, Ediacaran, although no definitive fossils from that period have yet been discovered. Since the earliest trace fossils also date to the earliest Cambrian, a much earlier origin of the phylum is considered unlikely. In the early Cambrian Maotian shales of China, and the Middle Cambrian Burgess Shale of Canada, arthropods are already diverse and well differentiated, represented by numerous species and individuals. The stem group of Panarthropoda is most likely the Lobopodians, also called Tardipolypods, an extinct group of worm-like animals with legs that disappeared in the Ordovician. In terms of body organization, though not necessarily ancestry, they resemble modern velvet worms, Onychophora. Primitive forms like Aschia show a segmented body with repetitive structures such as ringed legs but lack clearly defined true segments. On an unsegmented head, two long lateral appendages with stiff bristles are present, likely used for feeding. These are the only differentiated body appendages. Sclerotized plates were absent from both the trunk and the legs. Since then, a whole zoo of sometimes bizarrely modified Lobopodians has been discovered, which lived around the same time. Some of them show sclerotized plates or spines, and a primitively segmented head with one or two pairs of appendages. Examples include Onychodictian and the famous Hallucigenia. One species, the cactus animal Diania cactiformis, had limbs with jointed sclerites resembling the legs of arthropods. Fossils interpreted as stem group representatives of true arthropods, such as Fuxianhuia, Canadaspes, or Shankuya, display a number of new features compared to Lobopodians. The mouth shifts slightly ventrally from the anterior end of the body. At the front, a head composed of two segments is protected by a dorsal sclerite, tergite. The first segment bears a pair of stalked eyes, and the second bears sclerotized, jointed appendages that may resemble antennae or legs. The rest of the body is fairly uniform and segmented, with each segment protected dorsally by a strongly sclerotized plate, tergite. On the underside, stubby or leaf-shaped legs are present, usually weakly or not at all sclerotized, which allowed the animal to move across the seafloor in a swimming walking motion. Leaf or lobe-shaped appendages on the outside of the legs likely served as gills. More advanced forms show a head shield that covers not only the eye and first appendage segments, but also 3 additional segments, although their appendages do not differ significantly from the rest of the trunk limbs. This 5-segmented head is considered by many researchers to represent the basic body plan of EU arthropods. Whether fossils with this structure are to be interpreted as stem group arthropods, or as more derived representatives of specific arthropod lineages such as Crustacea or Chelicerata depends critically on how the anterior appendages and their segments are interpreted. Most researchers view the great appendages of many fossils, such as those of Liancoilia or Yohoia, leg-like, heavily bristled mouthparts likely used to grasp prey, as homologous to the chelicerae or first antennae of living arthropods. Following this interpretation, most of these forms can be considered stem group chelicerates. However, some researchers homologize these appendages with the antennae of Onychophora, which are located on the foremost head segment, the one that gives rise to the protocerebrum, the frontmost brain region, and thus do not directly correspond to the antennae or chelicerae of arthropods. Accordingly, these fossils are placed by those researchers in the stem group of all arthropods. Chylynxia was a 5-eyed, shrimp-like animal about 5 centimeters long that lived around 518 million years ago. With distinctive features, especially its anterior appendages, it is considered by some scientists to bridge the gap between Anomalocaris, Radiodonte, and Deuteropoda, true arthropods, and may represent the first true arthropod or at least the evolutionary root of the group. The stem group of crustaceans is already known from the early Cambrian, thus existing alongside Lobopodians. Particularly informative are body-preserved, phosphatized fossils, the so-called Orston fossils, which are only micrometers to millimeters in size, but reveal extremely fine anatomical details. Stem group representatives and members of the modern crustacean crown group are morphologically very similar, differing mainly in the structure of the head, which can consist of varying numbers of segments. The antennae of species like Oelandocaris were likely used primarily for feeding, much like the great appendages. In addition to forms that lead toward modern arthropods, numerous other arthropod lineages emerged in the Cambrian that have since gone extinct without leaving modern descendants. The most famous of these are the trilobites. Before the discovery of the rich Cambrian fossil deposits with preserved soft tissues, trilobites were the only fossil source for understanding early arthropod evolution. As a result, they heavily influenced earlier conceptions of the arthropod stem group. Today, trilobites are considered to be already highly derived, and in many respects, morphologically advanced special forms. Their traditional grouping with chelicerates into a taxon called Arachnomorpha has recently been questioned with strong arguments. Another famous side branch is the Anomalocaridids. Anomalocaris is one of the most iconic fossils from the Burgess Shale, and its relatives, reaching body lengths of up to 2 meters, were the largest predators in the Cambrian seas. Another side lineage, the Thylacocephala, went extinct at the end of the Cretaceous period. They have been found, for example, in the Solnhofen limestone deposits. Cambrian precursors such as Isoxes were among the most common free-swimming arthropods in the sea at that time. A further group, the Euthycarcinoidea, remains completely enigmatic despite well-preserved fossils. They have been linked to nearly every major arthropod lineage, yet their true relationships remain unresolved. If the interpretation of Cambrian trace fossils found in North America is correct, they may have been the first terrestrial arthropods, Fossils of primarily terrestrial hexapods and myriapods are abundant and sometimes excellently preserved, but they shed little light on the origins of these classes. The oldest fossils of both groups come from the same site, the Devonian deposits of Riney, Scotland. The animals found there were already terrestrial and had a largely modern body structure. Fossils of stem group representatives, e.g., marine forms, are unknown. All fossils interpreted as such are either questionable or have been definitively reclassified. That's it for now. In the next episode, we'll still take a closer look at Kingler. Feel free to subscribe